bench to bedside really implies the analysis of a particular idea. So the idea behind a scientific discovery, then taking that into the laboratory, that's the bench part, where you do fundamental experiments to test out either a hypothesis, a scientific thought, an idea, or to test a novel creation of a diagnostic, for example. And then ultimately, if you're designing, for example, a novel drug for a particular disease, this has to be tested. And this may be tested in experimental animals, but ultimately will have to be tested in patients for patient benefit. And that's the bedside concept. This also applies, of course, to development of novel diagnostics. So in the case of novel diagnostics, you would have the idea for the diagnostic, you would test out all of the reagents and the different conditions for running the test in the laboratory, and then you would take that into the clinic, to the bedside, by collecting clinical samples and testing them using your novel diagnostic. So from the bench to the bedside.
Going back into the lab was a great privilege, actually, and I really, really enjoyed it. But it took a lot of work to make sure that we are COVID safe and COVID ready for our class 3 labs. And a lot of people had to work very hard to make this happen. So I'm very, very grateful for everyone's help and support. But we got to the stage where we were permitted to culture the virus, and it was, I know a lot of people probably don't want to hear this, but it was just such a lovely thing to grow. So compared to the hepatitis viruses that I'm used to growing in the lab, that are growing very difficult, with great difficulty in vitro, the coronavirus was a breeze. It would infect very easily our target cells, and it would spread very nicely. And this is great news for research, really, because this way you can develop your assays very quickly, you can validate them, and you can test antivirals, you can test disinfectants, and you can progress your research towards a therapy.
Before coronavirus hit, my main interest was understanding how people's immune systems recover after having chemotherapy, mainly for blood cancers. And we were in the sort of early stages of developing a fairly comprehensive research program to look at that in more detail. Now, we've obviously had a pandemic that our immunology research has contributed to in understanding the risk that healthcare workers face when caring for patients in the hospital and elsewhere. And that has really sort of transformed what our clinical immunology service has been doing on a day-to-day basis.
So my contribution has mainly been in the early stages was around helping develop the antibody tests, and we now work together as a fantastic team with people like Alex Richter and with Mark Drayson and many others in developing a test that focuses on how to identify people who've either been recently infected or who have relatively modest responses to the virus. So all of this was a massively accelerated program. What would normally take a few years, we achieved in a few weeks. And this was not least in part because our ability to work with the Binding site, who've been awesome partners in this, and also just the natural collaborative environment that exists in Birmingham. So we were always working together. We've always had a very good working relationship. It's one of the great strengths of here. but every time we came to a door, we found that door to be open, and that just accelerated research.
In the early stages of the pandemic, we recognized that the tests that were available for testing for antibodies were perhaps not the greatest tests, and we went back to the bench to see whether we could do better. And that involved some broad collaborations with scientists across the university, and also scientists elsewhere who are experts at making protein, particularly with the University of Southampton. And really, the first thing we did was to try and just make a better test, and that involved a lot of work, a lot of gathering samples from people who we knew had COVID, and putting that all together into a brand new test, which has since sort of been commercialized and is now available for people to buy.
Very early in the pandemic, we had to stay open as a department in the university because we are an NHS-facing laboratory, and so we were key staff and we had to keep working. We realized very early on that there was a real need for testing. If you remember, the government's rollout of both antibody and PCR testing for the virus didn't happen until much later in the spring and the early summer. And so we decided to use our expertise in diagnostics and basic immunology to build our own COVID antibody test, which is what we've been working on. And this required a really significant collaboration and a pulling together of ourselves in clinical diagnostics and the basic scientists in our institute, in particular Adam Cunningham and his group, and then also our commercial partners in this, the binding site. And it's how we've all worked together with all the university legal communications teams, IP teams, to launch a product from conception to CE marking and launch in 4 months.
We're at both ends. We're both at the lab and basic science end, but we're also out there at the coalface in the NHS. And throughout the whole pandemic, we've been engaged with what's happening with patients and with NHS staff, with dentists, whole cohorts of people, getting literally thousands of samples of blood and also other things like saliva, so that we can help develop our tests, but also apply those tests to find out what was happening in the pandemic. So, for instance, back in March and April, when we were just starting out learning what's going to happen, we were able to find out how many healthcare workers had already been infected. And we're greatly surprised that even before patients with COVID were admitted to the hospital, many of the healthcare staff had actually already been infected. I guess at home, out in the community, and between colleagues. And then following up those individuals, now we're 6 and 9 months down the line from those early days, we can see what's happened to their immunity to the SARS-CoV-2 virus. How has it evolved? How has it matured? Is it maintained? These really important questions.
One of the most rewarding things that I've been doing during this pandemic is being able to support the work that our clinicians are doing here in Birmingham. We are able to get blood from patients that have had COVID immediately following infection, and months later, we are able to detect antibodies in those samples and see how people respond to the new coronavirus. And this is work led by Alex Richter here in Birmingham. So the clinical labs have developed really sensitive assays to pick up those antibodies to the virus. But what we would really like to find out is if these antibodies are functional. So can they neutralize infection? And this is where my lab comes in. being able to culture the coronavirus at category 3 labs means that I'm able to test our antibodies from our patients to see if they're able to neutralize infection. And this is not a surrogate system. This is using a fully replicating virus. So we can assign functionality to the antibodies with our assays in a matter of days. So it has been really interesting because we didn't know what to expect from this new infection. We didn't know whether people were able to mount a strong enough immunity to the virus. But now we appreciate that we are able to generate antibodies that are potent neutralizers and they can persist for longer periods of time.
The COVID-19 pandemic has really brought out the very best in people in our institute. There are people working on fundamental immunology. We have clinical immunologists who are looking after patients. We have teams of people working in the laboratories doing clinical immunology testing and developing novel diagnostics. And this year, in the face of this pandemic, it became clear that there was a real need to develop a true diagnostic test for the antibodies against this particular virus. And it's been incredibly gratifying to seeing all the people, too many to name, one by one, from the very fundamental immunologists through to the clinicians, the patent lawyers, the business engagement people, through to our colleagues in the university who've been communicating all our successes so effectively. And finally, to the wonderful team at the binding site in Birmingham, without whom we wouldn't have been able to take this test literally from the bench to the bedside and onto the market.